A warm welcome to my YouTube channel, Livestock Products Technology. Today I am going to talk in a very new and interesting topic, the development of skeletal muscle. This is of course with reference to meat science. So this topic is very important for meat science and for the animal science. Normally we discuss so many different aspects of meat, but this topic always remains less focused. Actually, this is one of the foundation about the formation of meat, because muscle is one of the major aspect about meat. That's why today we will discuss about the genesis of muscle. So it starts with fertilization, zygote, the embryonic development and embryonic differentiation, and then about the differentiation of myogenesis. These are all about the prenatal aspect, and then at the postnatal aspect we will see how the growth and development of the muscle happens and what are the different aspect and relation between the fibers and the number of fibers in the muscle. And finally we will see the important factors in the growth and development of skeletal muscle, like gender, species, the the genetics aspect, the breeding aspect, the nutritional aspect, and some of the growth factors, etc. As I mentioned, we are talking about the growth and development of muscle in the context of meat science. So in this case, growth and development of skeletal muscle is one of the most important aspect for animal science or meat science. So if we think about meat, the major part of it is made up of muscle, and the muscle is made up of muscle fibers. Hence, we will be discussing mainly here about the growth and development of muscle fibers. Though in the muscle there will be fat cells, connective tissue, the blood vessels and nerve fibers, but the major part of the muscle mass is made up of muscle fibers. So therefore, largely the number of muscle fiber and size of the muscle fiber is important to make the muscle mass and meat. So we will discuss mostly about this development of fiber and development of muscle, though there are many other aspects that we are not much interested, because our focus is about the meat. In case of any animals and the muscles, the greater number of muscle fibers of moderate size produce a higher quantity and quality of meat. So if a muscle has got more number of fibers with a somewhat higher diameter, that makes the more quantity and quality of meat. During myogenesis, the extent of muscle cell multiplication largely determines how many muscle fibers are formed. So myogenesis is very important to get the number of muscle fibers. The number of muscle fiber is mainly determined by genetic and environmental factors, and which influences the prenatal myogenesis. So this myogenesis happens before birth, that is prenatal. Whereas after birth, that is the postnatal growth of skeletal muscle, happens mainly by increase in length and girth of the muscle fibers, not by number. That means after birth, the length of the fiber and the diameter of the fiber can increase and thereby increase the muscle growth. But the number of fibers generally rarely increases, or only sometime it can happen. Now we will see the very basic aspect of embryonic development, that is embryonic differentiation. So during fertilization, the sperm and egg unite to form zygote, as we all know. The zygote undergoes cell division and becomes morula with 16 cells, and in uterus, morula differentiates to blastocyst. So blastocyst differentiates to inner cell mass and outer trophoblast. The trophoblast cells utilize the active transport of sodium ions and osmosis of water to form a fluid filled cavity known as blastocele. Now after 3 weeks, this blastocyst transform into gastrula, which will have three different aspects or three layers, we can say, that is endoderm, mesoderm and ectoderm. So these three layers gives the origin for different organs or different system, which we can say organogenesis. Here in this diagram, once again, we can see the stages of growth and development of the embryo. The egg, when it is fertilized with the sperm, it forms zygote, that is a single cell, and there it divides to 2 cell, then 4 cell, 8 and finally to 16 cell morula, and from morula there is formation of blastocyst, and from the blastocyst there is the formation of three layers of ectoderm, mesoderm and endoderm. These three layers gives the origin for all the organs or systems for further development. Here we can see the prenatal differentiation. So from these three layers, the different systems origin, that is the ectoderm, from which mainly the skin and other development happens. Then there is mesoderm and there is endoderm. So the muscle cells precursors are originated from the mesoderm, we can see, and the other developments, of course, we are not much concerned. We are mainly restricting to discuss about the development of the muscle. Here you will see the further details about the prenatal myogenesis. So first thing is, during embryonic development, myoblasts develop from myogenic precursor cells which are of mesodermal origin. So the first origin for the muscle growth starts from the mesoderm, and that is the formation of myoblasts. So these cells are destined to enter the myogenic lineage and are able to proliferate and divide to establish a pool of myoblasts. Then this special signal, a special signal cause the myoblasts to exit the cell cycle, to stop dividing and to differentiate. So there is a special signal which stop the myoblasts to divide, and it undergo a different kind of development or growth. So they begin to express muscle cell specific proteins and finally fuse to form multinucleated myotubes. So this myotube is the actual beginning for the development of the muscle, first by formation of muscle fiber and then formation of the muscle. Here once again we can see the prenatal myogenesis. In the top we can see embryonic myogenesis and fetal myogenesis. There in the embryonic part we can see the early myotome, later myotome and embryonic myotubes. So that comes with myocytes, first myotube and primary myotube, and then we can see fetal myogenesis where the fetal muscles are grown by the formation of secondary myofibers. So this is the diagrammatically explained about the prenatal myogenesis. Here once again we can see the formation of muscle fiber. It starts from mesoderm, then it forms the myoblast, then myocyte, then myotube and then myofiber. As I explained earlier, some cells are specifically destined and they becomes the myoblast, the origin for muscle, and their growth or development is stopped and there will be some kind of fusion and it forms myotube. And from this myotube the muscle fibers are formed, and with large number of muscle fiber it makes the muscle. Now we will see the development of muscle fibers at the prenatal period. So muscle fibers develop from two distinct populations. Firstly, from the fetal myoblast. So this is at the fetal myoblast level. So those formed during the initial stages of myoblast fusion are primary myofibers, as we have discussed earlier. By the fusion of myoblast cells, it forms the primary myofibers which provide a framework for the larger population of smaller secondary fibers. So primary fibers are produced at the early stage, embryonic stage from the myoblast, and then later from the development of fetal. After that, there is a formation of large number of small size secondary fibers. These are formed from fetal myoblasts during a second wave of differentiation. So these secondary fibers are formed from the fetus. The number of secondary fibers around each primary varies and can be over 20 in large mammals such as in pig. So these secondary fibers can be up to 20 in case of pig. Now we will see the another origin of the muscle fiber, that is the satellite cells, from the satellite cells. So these are another population of myoblasts which normally does not form fibers but stay close to the myofibers. These are termed as satellite cells, and they are able to divide and serve as the source of new myonuclei during postnatal growth. So during postnatal growth, sometimes they can produce the fiber. They contribute to growth of the fibers and also participate in regeneration process. So particularly during postnatal period after birth, whenever there is a critical situation or for regeneration, these satellite cells can produce fibers, particularly for regrowth or damage, etc. in such situation. So in the diagram we can see that myonuclei, there is satellite cell, and there is the multipotential stem cells or pluripotent stem cells from which the myoblasts are origin, and then myotubes are formed. Now we will discuss about the postnatal growth of muscle, that is after birth. The postnatal fiber maturation seems to be more dependent on other factors such as innervation, contractile activity, growth factors, hormones, and nutrition. So all these we are going to discuss in details later. So during postnatal growth, the increase in skeletal muscle mass is mainly due to an increase in muscle fiber size. So at this stage, the number of fibers do not increase normally. So when the increase in number of fiber, it is called as hyperplasia, whereas increase in size of the fiber, that is called hypertrophy. So hyperplasia happens in the prenatal period and hypertrophy mainly happens in the postnatal period. So both of these together makes the growth of muscle in case of making the meat. So this process is accompanied by the proliferative activity of satellite cells, as I told previously, which are the source of new nuclei incorporated into the muscle fiber. So there is second category of origin for muscle fiber, that is called satellite cells, and from the satellite cells also some new muscle fibers are formed, sometime in cases of such situation. Now after birth, total muscle fiber number has been reported to remain unchanged. Generally, the number of fibers remain unchanged except in some special cases in case of mammals. So each fiber can grow in diameter, that increase the size of muscle, and that is called hypertrophy. Here once again we can see the development of muscle fibers through this diagram. In the left top corner we can see the myoblasts at the embryonic stage. They fuse and form the myotubes, and from which the muscle fibers or immature muscle fibers are formed. These are all the prenatal. There is of course the satellite cell, which is the second origin of muscle fibers. Then at the postnatal stage, all these fibers can grow further and their diameter can increase, and that makes the higher size of the muscle or growth of the muscle. So that is hyperplasia. Whereas prenatal, there is multiplication of fibers, and that is called hyperplasia. Whereas when the diameter is increased, that is called hypertrophy. In continuation about the postnatal growth, in case of feces, they can have hyperplasia throughout life, and in later life, hypertrophy becomes increasingly important to growth. Hyperplasia after birth reported in rodents, chickens and pigs. Though I have mentioned earlier that postnatal life there is no hyperplasia, but there can be some amount of hyperplasia reported in rodents, chicken and pigs. In most studies, the fiber counts were done on histological transverse section. By making the transverse section, fibers can be counted whether it is increasing in number or not. The growth during first few days of postnatal life, that is just after birth, the growth is faster, that is by the maturation and elongation of the existing myotubes, that is the fibers, they grow by length and by diameter, so that makes the faster growth than by the new fibers. So new fibers are comparatively less or slow. Here we can see some more details about the postnatal growth from studies, especially in pig. So in case of pig, fiber formation is established between 70 and 95 days of gestation. So the fiber formation in the prenatal period is established between 70 and 95 days of gestation. Initially, increase in fiber number shortly after birth are observed in pig muscle. So after birth in pig muscle, some increase in number of fiber has been reported. The muscle fibers grow in size towards a plateau, whereas fiber number remains almost constant. So this is another observation. And then some decrease in fiber number with aging have also been reported. With aging, the number do not increase, sometime it can decrease. So these are some of the studies on the number of muscle fiber growth and other aspect. Now we will see the relationship between muscle fiber number and size in the muscle growth. The number of muscle fibers markedly determines the capacity of postnatal muscle growth. So we are discussing about the growth of muscle after birth, and this growth largely depends on the number of fibers, and this number of fibers are generally produced before birth. On the other hand, postnatal fiber hypertrophy mainly determines postnatal growth rate, but it is limited by physiological and genetic reasons. So there are several factors of physiological and genetic, and the postnatal hypertrophy that makes an important point for the growth of the muscle. So postnatal muscle fiber hypertrophy is inversely correlated with the number of muscle fiber within a muscle. Means if a muscle has got large number of fibers, then the growth of each fibers will be less. Whereas in a muscle, when the number of fibers is very high, then growth of each fiber will be less. When the fiber is less, growth of each of fiber will be more. So it is inversely proportional. Now we will understand about the factors affecting growth and development of skeletal muscle. So the first factor is the species. So this growth and development can vary between different species, which we are going to discuss all these factors in details. The second factor is the gender or sex, that is the difference between male and female. Then there is a factor of genetics and breeding. So gene has a control over the differentiation and growth of muscle, and we can manipulate this by selection and selective breeding. Then the nutrition is one of the major factor, and it can be in a two aspect like prenatal nutrition and postnatal nutrition we are going to discuss. Then there is physical activity. So use of muscle can help it grow more, that is by exercise or physical activity. And lastly, the growth promoting agents. So there are so many growth factors, growth promoting agents and hormones which has an important role in the growth and development. So we are going to see all these things one by one. So among the factors, first is the species. So here the species specific differences in muscle mass are primarily due to differences in the total number of muscle fibers. So different species has a different total number of muscle fiber, that makes a major difference in the muscle mass. So for example, in case of pig, longissimus dorsi muscle contains 6 to 7 times more fibers than in rabbit, that is the rabbit longissimus muscle, but the diameter there is only 2 times larger in case of pig than rabbit, whereas number is 6 to 7 times more. So ultimately the muscle mass is very high in case of pig than the rabbit. Similarly, another example, in case of cattle, it has got 3 to 4 times more number of fibers than pigs in a particular muscle, 3 to 4 times more number. So naturally it makes bigger mass. However, the size is also controlled by the fiber diameter, where the fiber diameter it is more in case of pig than in case of cattle. So all these things together makes a difference in the muscle mass between the species. The second factor is about the gender and sex. So as we all know, the male body is always more muscular. So intact males mostly exhibit larger muscle fibers than females, or even more than the castrated males. Although in case of pig, sometimes partially larger fibers have been reported in case of female than male. So in case of pig there is not much difference, or sometimes it is more in case of female, but in general in most of the species the male has got the better muscle, mainly due to larger muscle fiber. So these differences is mainly due to the control by testosterone hormone. The number of muscle fiber and the size of muscle fiber is larger in case of male, mostly by the testosterone. In the experimental basis, the testosterone treatment in later postnatal period could stimulate the muscle fiber hypertrophy. That means whatever fiber is there that has increased the growth or diameter, which has been treated with testosterone. So it has a direct or indirect stimulation to the satellite cell proliferation and muscle protein synthesis without increasing the number. So treatment with testosterone can cause hypertrophy by stimulating the satellite cell and producing more muscle protein, and thereby causing the hypertrophy, though the number remains same. So another factor is the genetics and breeding. Particularly nowadays we have selective breeding for improving performance in some particular aspect in the farm animal. First of all, the muscle growth is controlled by several genes. Some genes promote the growth of muscle, whereas some gene has a negative impact in the growth of muscle. So one of the common gene is myostatin, which acts negatively and prevent the growth. So if we design some kind of breeding where some particular breed has got higher muscle growth, means it has got less influence of this particular gene and allow the muscle to grow. So whether and to what extent a biological trait is inherited and can be changed by selection largely depends on its genetic variability, heritability, and genetic correlation to the criteria used in the selection. So in a particular selective breeding, we can select a gene which helps in improving that particular criteria, and we have to see the heritability, how it is transferred to the next generation. So that is very important for application in the selective breeding. Within a species of farm animals, one of the most important factors that influence muscle fiber number and size, that is the genetic factors, and selection and breeding with the aim of improving animal performance. So one of the important criteria is the number and size of fiber, based on which the selective breeding is done for more meat production. Now we will see the effect of nutrition in muscle development and growth. So adequate nutrition is essential for normal skeletal muscle development. It is quite well known that nutrition plays a very important role, and this we will discuss in two parts. First is the prenatal nutrition. So prenatal nutrition means the nutrition of the parent. So prenatal undernutrition in pig has been shown to be related to lower muscle fiber number, as evidenced by the comparison of small with large weight piglets. So it has been observed by undernutrition, the small and large weight in case of piglets. Similarly, in contrast, doubling of maternal feed intake of sows during gestation tended to increase semitendinous muscle fiber number. So in case of pig, by feeding double to the mother, it has been observed that semitendinous muscle fiber number has increased in the semitendinous muscle. So precisely, the prenatal period of muscle development is much more sensitive to nutritional deficiencies because this period includes the muscle fiber formation. During this prenatal period only muscle fiber is formed. So nutrition at this stage is very, very important. Now we will see about the postnatal nutrition. The malnutrition during postnatal growth reduces body weight including skeletal muscle weight. So it is quite common after birth. If the malnutrition happens, the overall body weight growth or muscle growth will be reduced. So feed restriction both in quantity and quality, that is lower in protein and energy, has been reported to decrease the muscle fiber diameter. If the feed is of poor quality, limited in protein and energy, the muscle fiber muscle fiber diameter get reduced. So naturally the muscle growth is affected. So here during the early stage of the postnatal period, just after birth, that is the elongation of existing myotubes, which leads to an increase of apparent fiber number during early postnatal growth, cannot be completed when the animals are undernourished during early postnatal life. So at the early stage after birth, if the nutritionally they are underfed, then the elongation of myotubes will not happen. So number of muscle fibers will not come. So that leads to poor growth of muscle. Here is the another important factor, that is the physical activity, which has a relation with the muscle growth and development. So physical activity is an important factor that influences the adaptation in the muscle. Different housing system allow different degrees of physical activity for farm animals, or if there is a large distance to walk and for grazing, that gives a physical activity and improves the growth of the muscle. However, targeted exercise programs are not commonly applied in animal production. Immobilization results in a decrease in muscle mass, which is accompanied by decrease in muscle fiber cross-sectional area, that is the diameter will be reduced and the fiber number also to some extent. So when the animals are fully immobilized. So that's why the exercise or movement or grazing in a large field, long walk, helps in physical growth of the muscle and better meat quality. Now the last factor on growth and development of muscle is the growth promoters and hormones. Growth is known to be regulated substantially by the neuroendocrine system as well as paracrine and autocrine actions of hormones and growth factors and the influence of environmental stimuli on the individual. So growth promoters, growth hormones are very important for muscle growth. Based on this knowledge, growth promoting agents that can influence muscle growth in farm animals are growth hormone, beta agonists and steroids have been studied. So we are going to see the effect of these three growth promoters and hormones, that is growth hormone, beta agonists and steroids. So among these, first we will discuss about the growth hormones. Postnatal application of growth hormone increases lean growth and decreases fat deposition. Means it will increase the growth of lean muscle with more protein and decreases the fat deposition. The response to growth hormone in pigs largely depends on the dosage, sex and breed. So it has been experimented in pig and found to have depending on dosage, sex and breed. The second factor in this growth promoting agents are the beta agonists. They act on the beta adrenergic receptors. The growth promoting effects on muscle by stimulating fiber growth are also known to be exerted by beta adrenergic agonists in various species. So they are also called as repartitioning agents. So they can influence the use of energy towards more protein synthesis and less fat synthesis. The beta adrenergic agonists are able to increase lean growth and to decrease fat deposition in cattle, sheep, pig and poultry. So these growth promoters, they can use the nutrients towards making more protein and depositing less fat. So this is the another factor under the growth promoter or hormone. In this, the third one is the estrogens and androgens. And of course, we have discussed about the testosterone earlier, which plays a major role in case of male, particularly in the hypertrophy of the muscle by increasing the diameter of the muscle fiber. So these steroids are anabolic hormones whose growth promoting effects are well known in cattle, whereas limited effects are known from swine. So this effect of anabolic steroids are more in cattle than in swine. However, studies on the effects of steroids on the micromorphology of muscles are rare. So finally, we can conclude about these hormones that hypertrophic response in postnatal muscle to growth hormone or beta agonists or steroids is based on muscle fiber hypertrophy. So the main response is due to the increase in the diameter of the muscle fiber, leading to the more growth of the muscle, but not by the number of muscle fiber increase. So the multiplication of muscle fiber do not happen with these growth factors, only it can increase the diameter, so that is by hypertrophy. So now we are at the end of today's lecture. Today our topic was the development and growth of skeletal muscle with a context for meat science. So here we have discussed what is the importance of growth of muscle and the production of meat in terms of meat science or animal science. First we have discussed about the development of embryo and embryogenic differentiation and the growth of muscle fiber. We have seen how some myoblasts get segregated and their multiplication is stopped and they get fusion and forms the myotube from which the muscle fibers are born in the prenatal stage. And later we have seen the postnatal growth where the number of fibers do not increase much. Mostly there is an increase in fiber diameter, and how it is happening and how the relationship of muscle fiber diameter and muscle number and the muscle growth is related. And then we have discussed in details about the different factors that affect the growth of muscle, like the species, sex, breed, or the physical activity, and the genetic aspect or breeding aspect or selective breeding. And we have seen the effect of nutrition, particularly prenatal and postnatal nutrition, how it influences the growth of muscle, particularly fiber number or fiber diameter. And finally we have seen the effect of growth promoters and hormones like the growth hormone, the beta agonist and the steroid hormones plays important role in the muscle. So in brief, I have discussed here about the growth and development of muscle in relation to meat science, which makes a foundation for understanding about the meat production from the meat animals.